Hello friends, this video on Principles of Inheritance Part 22 is brought to you by examfear.com. No more fear from exam. So now we will talk about the behavior of chromosomes during meiosis. We are more interested in meiosis because meiosis is the type of cell division which is involved in the process of gamete formation. And gamete formation plays an important role in the process of inheritance, because inheritance is something which is related to sexual reproduction. And in sexual reproduction, gamete formation is the process by which it is initiated. So meiosis play a major role there. So let us quickly see what happened during meiosis and how the chromosomes underwent a change during meiosis. So meiosis, it is a specialized type of cell division which reduces the chromosome number by half, and that is why it is also known as the reductional division. So when I say chromosome number is reduced by half, what I mean is if you start with a diploid cell, you can end up getting haploid cells. So that is how your chromosome number gets reduced. So diploid cells will have two sets of chromosomes, and from that you end up getting haploid cells with one set of chromosomes. This occurs during gametogenesis in plant and animal cells. Now, why meiosis occurs during gametogenesis? Because the gametes, in the process of gametogenesis, what happens? Gametes are being formed, and gametes are haploid cells. So basically, we want to form haploid cells. So we need meiosis. So in case of human beings also, the gametes are the sperm and the ovum. So they are all haploid cells. So in order to produce these cells, meiosis has to form. Even in Mendel's experiments, we saw, right, how gamete formation occurs. So for gamete formation, you need meiosis. Now the process of meiosis involves two sequential cycles, and they are meiosis 1 and meiosis 2. So the meiosis 1 is the reductional division where the actual decrease in the chromosome number take place. So at the end of meiosis, 2 haploid cells are produced from one diploid cell. So you start the process with one diploid cell. After meiosis 1, you are left with 2 haploid cells. After meiosis 2, you are left with 4 haploid cells. So basically from one diploid cell, you are able to produce 4 haploid cells. So meiosis 2 is almost like an equational division. I mean, it is almost similar to mitosis, because mitosis is an equational division where the chromosome number remains the same. So meiosis 1 will reduce the number of chromosome to half, and meiosis 2 will divide it further. So you will get 4 haploid cells from a diploid cell. So that is what you will get. So if you have one diploid cell, at the end of meiosis 1 and meiosis 2, you will get 4 haploid cells. So let us look at the overall process of meiosis. This is how the overall process looks like. So if you see, this entire part is meiosis 1. So this is meiosis 1, and this entire part is meiosis 2. So each meiosis in turn has 4 stages, that is prophase, metaphase, anaphase, and telophase. Now I will not get into the very much detail of meiosis. If you want to learn it in detail, you can refer the lesson on cell cycle of class 11. There I have discussed meiosis in detail. However, I'll give a quick review so that at least for now you are able to understand the next concepts which we are going to talk about in chromosomal theory of inheritance. So this process basically starts with prophase 1, and in prophase 1, what you see, a lot of major significant changes take place during prophase 1. So if you see in prophase 1, the homologous chromosomes become visible. So here you can see this is one chromosome, this is another chromosome. So these two are the two homologous chromosomes. I hope you can see this is one, this one is one, and again this is another one. So you have one set of homologous chromosome, again here you have one set of homologous chromosomes. So here the homologous chromosomes start getting visible. So let me try to write it here, that will make things easy. So what happens during prophase 1? So initially, how was the homologous chromosome? This is how the homologous chromosome look like. So these two are homologous chromosome. Then what will happen? The homologous chromosome the chromosome duplicates to form the sister chromatids. So now you have duplicated homologous chromosomes, somewhat like this, right? In this prophase 1, it is also seen that the homologous chromosomes start to pair up. Now what do I mean when I say they start pairing up? So this is one homologous chromosome. and this is another homologous chromosome, right? So if you see, they have started pairing up. And this is very, very important. So these sites at which they start connecting with each other, or they start getting linked to each other, these are known as the sites of crossing over. What happens at the sites of crossing over? They actually change a small part of each other. That is what happens as a result of this crossing over. So at these two regions, they are called crossing over. At these two regions, there some part of the first chromosome get exchanged with the second chromosome, and some part of the second chromosome gets exchanged with the first chromosome, and this process is known as recombination. So towards the end of prophase 1, what happens is the first homologous chromosome looks somewhat like this. So if you see, the cornermost portion have got exchanged, and the second homologous chromosome looks somewhat like this. So if you see here, the entire chromosome is white, but these portions which underwent crossing over, they exchanged some of their parts. So as a result, what happened? So you basically now have two homologous chromosomes, but some of their parts have been exchanged. Now what happens if a part of the chromosome has got exchanged? The chromosome contains gene. So that means the gene which was present in this part got exchanged with this part, and the gene which was present in this part got exchanged with this part. So basically the genetic material is getting mixed up. So there is some blending which is happening inside between the maternal traits and the paternal traits. So it is quite possible that, let us suppose, the gene for hair color and was present here, and that gene for hair color went here, and it got blended or mixed up. So you might find new traits due to crossing over, right? So I am not getting into the detail that how it is formed, when it is formed. For that you can refer those videos, but just try to understand this, that when crossing over takes place, basically what happens is that some of the traits, there is some mingling or some mixing up of some portion of the chromosome, and every portion of the chromosome contains some genes, and those genes control some traits. That is why in a child there might be some new characters which are not exactly present in the same way in any of his parents, and this happens due to crossing over, right? So this is one important thing which takes place here, and this is going to be one important point when we talk about the chromosomal theory of inheritance. So this is how chromosome plays a very important role. Now what happens in the next stage, that is the metaphase. In metaphase, microtubules start to form. So here you can see a network being formed, that is called the spindle fiber network. So these microtubules gradually tend to form the spindle network, and as a result, all the homologous chromosomes are aligned at the equatorial plate. Now how do they align at the equatorial plate? They are aligned due to the continuous forces which are exerted on these tetrads by the spindle fibers. So the spindle fibers attach to the homologous chromosome. So here you can see the spindle fibers are attached here, and they all align at the equator of the metaphase, and this is known as the formation of the metaphase plate. So this is called metaphase plate because a plate-like structure is formed along the equator at this stage. Now what happens in anaphase 1? So in the next phase, the homologous chromosomes separate. So once they get aligned at the equator, then what happens? This spindle fiber tend to push it this side, this tend to push it this side, so both are separated away from each other. But still the sister chromatids are still attached. So if you see, so one homologous chromosome goes this side, the other homologous chromosome also moves this side, and on the other side you have again a pair of homologous chromosomes, right? And if you also see that here not both the same set of homologous chromosomes are going to one side. So one blue is going this side and one red is going this side. Similarly, one blue is coming this side and one red is coming this side. So basically when the cell will divide, so each cell will have combination. It will have one set of chromosome from one homologous pair, it will have another set of chromosome from the other pair, right? And the sister chromatids are still attached. So here you can see these are the sister chromatids. So they are still attached. So at the end of anaphase 2, anaphase 1, what happens? Telophase 1 starts. And what happens in telophase 1? So in telophase 1, cytokinesis starts. So in telophase, cytokinesis. And what is the meaning of cytokinesis? It means division of the cytoplasm. So the cytoplasm will start to divide. Once the cytoplasm divides, it will start developing the plasma membrane, and the proper two different cells will be formed. So two daughter cells are formed with half number of chromosomes. So if you see the initial cell, how many chromosomes it had? If you see here, it had, this is one pair of homologous chromosome, this is one pair of homologous chromosome, right? So it had two pairs of homologous chromosome. Right now, it just has one pair of chromosome. So the chromosome number has reduced to half. So you received two daughter cells. So two daughter cells, and these daughter cells are haploid. Right? So haploid cells are formed at the end of meiosis 1. Now what happens to each of these cells? Now each of these haploid cell will undergo meiosis 2. What happens in meiosis 2? It is very similar to mitosis. So basically here again spindle formation will take place in the prophase 2. So the chromosomes become contact and spindle formation starts to begin. Now once the spindle again attaches itself to the centromere of the sister chromatids, and then again the spindle and again the sister chromosomes will align themselves at the metaphase, at the equatorial plate, and it will form the metaphase plate in metaphase 2, and again due to the force exerted on both the ends, the sister chromatids will separate here. So in anaphase 2, what will happen? The sister chromatids will separate. So here sister chromatids will separate, and as a result you will be left with 4 haploid cells. So as a result, 4 haploid cells will be formed. So basically here in this case, the sister chromatids were still there. So this was just 2 copies of the same chromosome, something of that sort. Now they also got separated, but you can still see that due to recombination, some portion got exchanged, right? So these are the portion of the chromosomes which got exchanged, and these portions will actually give rise to some new traits in the offspring. So here in this phase, if you look at the anaphase 2, you can actually relate it to the principle of segregation, where for gamete formation, the alleles tend to segregate. So that is what is happening even here also, right? So as you see, the end result is that you started with 1 diploid cell and you ended up with 4 haploid cells. And during this process, the thing that makes meiosis very much different from mitosis is the changes which take place during prophase 1, that is crossing over, recombination. So these are the changes which make meiosis very much different, and as a result of which the cells which are produced as a result of meiosis, they are not exact identical clones of their parents. They show some new traits. They also show some differences from their parents, and all that is because of the process of crossing over which take place during prophase 1. Now, looking at the similarities between the behavior of chromosomes and the behavior of the Mendel's factors, an improved theory came up. Now, this theory doesn't come up on its own. There were quite a few scientists who started working on, who started to progress the work further on genetics. So they kept on performing more and more experiments. They tried to, now since cell division was properly visible now, so they started understanding the behavior of chromosomes, how do they behave, and then they found that whatever laws were given by Mendel, the behavior of the factors were quite similar to the behavior of the chromosomes. So now they started to connect the dots. They wanted to develop, or they wanted to connect the link between the factors, that is Mendelian factors, and chromosomes, and that is how they came up with the chromosomal theory of inheritance. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.